In this video, we're going to discuss the pathophysiology of Alzheimer's disease. In Alzheimer's disease, the brain actually shrinks in size. It atrophies. So on the left side here, you can see that you have two neurons represented here. You can see the axon and the dendrites. They look nice and healthy. They're reaching out. They're communicating with each other. There's nothing in between them that is causing any issues with communication in the synapse. And then in contrast over here, you can see the atrophied brain represented for Alzheimer's. You have the neurite retraction. You can see how these dendrites are kind of retracted back. You have neurofibrillary tangles intracellular, inside the body of the neuron. That's a hallmark of Alzheimer's, as well as amyloid plaques that are kind of gumming up the synapse. A place that you see disproportional atrophy with Alzheimer's disease is in the hippocampus. The hippocampus is the region deep within the brain that our memories are held in there, and are both long-term and short-term memories are located in the hippocampus. And you can see all the dark image there from the MRI, whereas on the normal, it's kind of filled out. You can see the substance of the hippocampus there. Another hallmark of Alzheimer's disease is impaired glucose metabolism. Our brain requires a lot of energy in the form of ATP. Most of that energy comes from glucose, and if glucose is impaired, some people have even coined this as type 3 diabetes, where you have insulin resistance inside the brain. So here would represent a brain of a person with mild to moderate Alzheimer's disease, where you want to see a lot of red. You don't see that as much on this side as compared to the healthy brain, where the insulin's coming in and there's plenty of receptors for insulin, so that the glucose can be pulled into the neurons, and ATP can take place, or ATP formation can take place. So in other words, you're starving the brain tissue of energy when you have this impaired glucose metabolism. There's actually a gene called the Alzheimer's gene, which is when you have an ApoE4 allele. There's 3 alleles that you can get. You can have an ApoE2, 3, or 4. The 4 is the one you don't want, 3 is kind of middle of the road, and 2 is the one you would like to have if you don't want to get Alzheimer's disease. And ApoE4 does quite a few things that are deleterious or bad to our brain. One is we have less amyloid beta clearance. So on that first slide, you remember seeing the amyloid beta that gums up the synapses between the neurons. You get less clearance. It plays a role in clearance, and with less clearance, you get more aggregation, as the picture shows here. You actually get more of those neurofibrillary tangles, the tau tangles that are inside the cell body of the neurons. You get brain atrophy, and with those amyloid plaques, you're going to have decreased synaptic function. You're not going to be the neurons aren't going to be able to communicate at the synapses as effectively. ApoE4 is actually a lipoprotein, and it carries fat and cholesterol, so you have decreased lipid and cholesterol metabolism when you have an ApoE4 allele, which I was unlucky, and I have one allele for. So fortunately, Dale Bredesen, a neurologist, has come up with some ways that you can prevent Alzheimer's when you have this allele. So I think it's important to check for this. The way I check for it is through Boston Heart Lab, which is just getting your blood drawn, but they look for a lot more information than just a typical one. And mine showed that I had one allele from one parent that was ApoE4, and then one was ApoE3. And even though it's bad news, now that I know, I can use Dr. Bredesen's protocol that is actually shown to prevent Alzheimer's from forming, and actually for the first time ever in research shown that you can reverse Alzheimer's once you have it. So it's a progressive neurological disease, and he's actually showing that you can regress it. So it's empowering to know that, and you can do 23andMe and find out. I did that with my son, and he also has it, thanks to me, obviously. There's probably some other ways, but those are the two ways that we found out in our family. And mitochondrial function, it decreases so up here in this right upper quadrant, impaired insulin signaling, impaired glucose metabolism, and decreased mitochondrial function are all going to reflect negatively on our ATP production. If the ATP is produced primarily in the mitochondria, and if the mitochondria are dysfunctional, that's going to make less ATP. If you can't get glucose into the neurons to actually go through the Krebs cycle and make ATP, then you're going to have impaired glucose metabolism and thus less energy. Insulin signaling, if you have resistance to insulin, it's going to do the same thing. Another thing that people like myself with the APO4 allele have to look forward to is blood-brain barrier leakage. The blood-brain barrier has all those tight junctions, those intercellular junctions that line the endothelium and really make it picky as to what gets into the brain and what gets out. And with the APO4 allele, it has a lot of issues at the blood-brain barrier level. Some things are leaking in that aren't supposed to. Some things can't get across that would normally. For example, we have a lot harder time of getting omega-3s from the peripheral blood into the brain. That inner leaflet of the blood-brain barrier causes some issues. So the only way Rhonda Patrick, she's got an interesting theory on this. She's found that phospholipid, through her research, phospholipid forms of omega-3s can actually make it across the blood-brain barrier. And there's not too many foods that have a lot of phospholipid omega-3s. There's a little bit in like salmon and different oily fish, but the actual salmon eggs, the caviar called salmon roe, have the highest concentration and percentage of phospholipid form of omega-3s. So every time I get sushi, I always get some salmon roe and choke it down because it is not too appetizing. Get lots of ginger and put in there and try to mask some of the taste. You can't really talk about the pathophysiology of Alzheimer's without talking about amyloid precursor protein. This is a protein you can see in the middle here, and it can go one of two ways. If it goes over here to the left and is cut into two peptides, that's great. That's going to signal to promote synapses, so it's synaptoblastic. If it ends up getting cleaved by a different enzyme at 4 different spots and makes these 4 different peptides, although this comes from the same progenitor protein, it has a whole different effect. So it's going to cause synaptoclastic bad signaling and cause the amyloid to build up and all that. So you can see there on the right, it builds up amyloid plaques when you get this. And the neurofibrillary tangles, it causes mitochondrial dysfunction, so you're not going to get your energy. Causes neurite retraction, so these dendrites, maybe the axons, the spines, they all kind of retract back where they're not going to communicate as well. So APO4 causes it to go more to the right, where it gets cleaved the wrong way. And there's a lot of things that can affect this. Different things like stress and thyroid hormone and testosterone and even melatonin. Different hormones can alter this balance favorably or bad in a bad sense. So this balance is important, and it's even more important to figure out how to push the balance to the good side when you have APOE4 going against you. So in the next video, we'll talk about Del Bredesen's protocol and what we can do to fight Alzheimer's disease. He's a neurologist that for over 30 years tried to find the magic bullet, but he found 36 things that help. And if you work to bring all those 36 things together, you can actually prevent or even, for the first time ever, reverse Alzheimer's disease.